So this is a schedule. And now so this is the schedule here. And you can see that we have kind of a fairly packed 11 weeks, but it should be fun, okay? And so at least my lectures will be all online. Tina, I think, will summarize her lectures and put them online as well, as will Sonia and Bhavana and Soumyashri and Rolf, okay? So let's start, okay? So I always start the course with this quote from Darwin, from The Origin of Species, and I'll actually read it out because it's always quite inspiring. It is interesting to contemplate a tangled bank, clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the damp earth, and to reflect that these elaborately constructed forms, so different from each other and dependent upon each other in so complex a manner, have all been produced by laws acting around us. These laws, taken in the largest sense, being growth with reproduction, inheritance, which is almost implied by reproduction, variability from the indirect and direct action of the conditions of life, and from use and disuse, a ratio of increase so high as to lead to a struggle of life, and as a consequence to natural selection, entailing divergence of character and the extinction of less improved forms. Thus, from the war of nature, from famine and death, the most exalted object of which we are capable of conceiving, namely the production of the higher animals, directly follows. There is a grandeur in this view of life, with its several powers having been originally breathed by the Creator into a few forms or into one, and that whilst this planet has gone circling on accord to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. So this sums up why I at least like how I think about developmental biology. I think of it as a way of understanding how all of these beautiful forms have come into being. And so this is a slide that is borrowed from Michael Richardson, who is who's portraying the kind of more realistic version of Haeckel's recapitulation theory. And so what you're seeing here from the left, the lamprey, a basal vertebrate, to us humans, from early in the top row, intermediate, and late. You're seeing that there's a stage at which all vertebrate embryos, at least, go through where they all look the same. They diverge soon after, and you start seeing their adult forms starting to appear. Okay, all of these words that I'm saying, form, divergence, are loaded with meaning, and may not be, and I'm not purposefully not defining them so clearly at the moment, okay? But basically, what we're trying to understand when we think about developmental biology is how we go from this round dot, an egg, on the left-hand side, to us, or this small dog here, or the bird that's flying in the sky outside your window. All of these are coming from one single cell. And in these lectures, we're going to try and explore how developmental biology is basically underpinned by many, many different concepts, many different fields that all work together to give you this understanding of how these different forms form from something formless. As a kind of treat for you, I'll show you my dog. This is the same dog here, who's much older now, but is one of the pinnacles of example of developmental biology, I believe. Okay, so is he going to move? Ah, he's moving. Okay. Okay. Sorry. This slide was actually taken by me maybe 22 years ago. So what you're looking at here is a chick embryo, and you're looking at the ventral side. This can you see my arrow? This cursor here. Is that visible?
Yes.
Yes.
Yeah. Okay.
Yes.
Good. So what you're seeing here is the this is going to be the future brain. This is a structure called the primitive streak, and we'll come to that much later. Okay, and it's churning out tissue, churning out one of the two of the layers of the embryo called mesoderm and endoderm. And what this slide is hopefully going to show you is that, and this is a time-lapse image, and it's showing you this emergence of form, okay? And as I mentioned, form is kind of this nebulous meaning at the moment, okay? It's not the final form, they're intermediate forms. And so I think what one trick you have to have as developmental biologists is to think in 4 dimensions. So we have taken a cell here in this region, and as the embryo develops, you can see that it's kind of converging into the midline. It's thickening, and this is forming the neural tube, which is going to form the central nervous system. This region here is going to form the heart, and you can see that it's now undergoing this very wonderful morphogenesis, these shape changes, these folding events, and now it's beating as it forms the final structure of the embryo here. Okay, so form in this context is an intermediate form, and when you start thinking of things in 4 dimensions, you know that ultimately this line here, this tube here, is going to form the central nervous system. This is going to be the heart. These blocks here are going to be the vertebrae and the muscle blocks in the embryo. Again, going even earlier, this is a zebrafish embryo. And I'll let this play first, and you can see that you have cell division, you have a kind of cell reorganization going on, and then the final form of the embryo is becoming more and more apparent. Here you can start seeing the eyes of the fish starting to form, the muscle blocks and the vertebrae forming, and here it's moving around in its egg, and very soon it will be free from this, it will hatch from this vitelline membrane. And so there we go. So 4 dimensions. What we were looking at, individual cells, thinking about their behaviors in toto, how individual cells behave in the context of the whole organism. And this is much more apparent in the next slide here, where Phil Keller, who's now in Janelia Farm, has done a light sheet imaging of the nuclei of the zebrafish embryo. And you can see that you have cell division, you have some morphogenetic events, as the whole of the embryo becomes more and more apparent. So this is, this to me is the beauty of developmental biology. We're looking at things in every scale, from the subcellular understanding of how these cell division events happen, how they're synchronized, to biochemical events to understand how patterning cues are set up, and to more tissue level events to understand how individual cells reorganize, and then to organismal events to understand what the organism is, what the animal or plant is sensing and seeing, how that affects its final form as well. So this course is an 11-week course, every Monday, Wednesday, Friday. It starts at 9:30, and we'll use this Zoom link that you all have, that's in the Moodle site, and it's on the Google Calendar, to connect at 9:30. It's taught by Tina Mukherjee, Sonia Sen, Bhavana, Soumya Shri, and me, as well as Arjun and Rolf as well. We'll have 2 or 3 assignments during this session, which will make up about 30 to 40% of the course mark. There'll be an end of course assignment as well that will make up the rest of the mark as well. There'll all be assignments. We won't have any sit-down tests. If we do, there may be some pop quizzes, but nothing that I think you can't handle. We'll have discussion classes, and we'll share literature and lecture notes on Moodle. So if you're not already on Moodle, please sign up. Okay, so are there any questions at this point? No? Okay. So if there are no questions, I'll carry on. So the first thing I want to discuss is actually what is developmental biology. I gave you some idea of what I thought it might be, but actually this is a much more difficult question than I realized. So there's over the past 20, 30 years, there's been a few definitions of developmental biology, from all of these very learned people. Here, the first definition: the process through which a single cell gives rise to a complex multicellular organism, is one definition. This here, the possible difficulties are that it excludes potentially several important developmental processes like regeneration, excludes many organisms such as unicellular organisms, which do undergo some level of development as well. So if you've ever looked at Colobacter differentiation, you understand that can be classed as development as well, and is adult-centric. It's the teleology is the drive to make an adult, which may not be particularly correct. Second definition put forward by Amundsen, Love and Pardue, is the elaboration of a new individual form. Again, it's difficult to define the notions of new, shape, or form. So what do these mean? Slack, Jonathan Slack in 2013 said it was the change of biological form through time. Again, this idea of form is a little bit ambiguous, as we're coming to realize here in the context of development and embryology. Form is a slightly plastic term. It's a 4-dimensional term. And so the change in biological form through time is maybe on the face of it correct, but it also doesn't incorporate, here one argument against this is that it's difficult to distinguish development from metabolism, for example, where changing metabolism over time could also be called development. Temporal change of organization along the life cycle. Here, organization was difficult to define. Biological readout of genomically encoded networks that determine how the organism is constructed seems like it's really a very narrow definition, and it ignores things like hormones, mechanical forces, environmental factors, and so on. Irreversible increase of complexity over time. Complexity? What's that? There are cases where development seems to be associated with a decrease in complexity. So Ascidians, for example, where during their metamorphosis, kind of their change into the adult form, they actually lose most of their nervous system and just are a sessile gut, or in the case of metamorphosis of parasitic animals. The restriction of possible outcomes, specialization, and increase in the levels of organization. And again, this is very dependent on how you view these particular terms. So something I try to get out of all of these meanings was that there's definitely this idea of the acquisition of the adult form. So this is kind of teleological. There's a purpose, if you want to call it that. There's a change as a function of time. And so that's very clear. There seems to be an increase in complexity, but again, we have these exceptions that seem to complicate things. But there's something else as well, and this I don't really know how to grasp and grapple with. So it should be said that if you had a look through the Stanford Philosophy Department's definition of, or their entry for developmental biology, one of the big things they talked about was the fact that there is no real, there's no theory for developmental biology. And so this is certainly true, and I don't think it matters too much. So I think developmental biology, I think, is just a bunch of lots and lots of different ideas and theories which are somehow coming together to give us this more holistic view of what's going on here. Okay, so I just want to add one more point to what is developmental biology, and I want to suggest that it's the process that enables a transition of an organism between the stages of its life cycle as well. And in this way, development becomes perhaps too broad. You know, if you're thinking about stages of life cycle, what does that include? I would say that it includes everything. You know, the growing once you're born, as you grow, as you undergo puberty, as you age, is all part of development. And so maybe this is showing my biases. I think everything is developmental biology. Okay.
Raj?
Yeah.
Yeah, so this last definition that you used,
Uh-huh.
I just wanted to ask if you think this will include the process of regeneration, as that might not be necessary within the life cycle of an organism.
Yeah, I think it does include the process of regeneration as well. And so regeneration would be a response to damage, the need to repair. And of course, it's not strictly part of the life cycle, but it's, so you're right, but it could be some aspect of the life cycle which does involve repair. I seem to have stopped sharing. Okay. Sorry, I've just seen the comments that the slides were blurred. Were the slides blurred?
Yeah.
Yeah.
I think, Raj, you might have ticked that option for optimize full screen sharing while sharing the screen. That causes this problem at times.
Okay. Which one? Optimize. Which one is it?
So when you press the button share screen,
Uh-huh.
you get share computer audio and another option for optimizing it.
Okay.
You have to untick it,
Okay.
and that might solve the problem.
Okay. Okay, let's see that. Okay. Thanks. Okay, how's that?
It's clear, much better.
Okay. Luckily, all of this is on Moodle, so you can download it. Okay. And see exactly what the words are. Okay. So the question,
Yes.
Is that okay?
Partially, I'm still not very convinced how it includes regeneration.
Yeah. So maybe this is definitely this is ridiculously broad definition. Yeah, and maybe it just says that I need to refine it even more. Okay. Yeah. So what do you say? What do you suggest?
It's kind of tricky. I'll think about this and let you know if I come up with something.
Okay.
But I think, for example, like our livers have the capacity to regenerate, but it might not be necessary during my lifetime. So my life cycle will not include that as a necessary part of it. So I think these things will get excluded if we use this definition.
Okay. Okay. Cool.
Yeah.
Yeah. Okay. Well, yeah. Need to put repair in there at some point as well, I guess. Okay. Nice. Okay. Any other questions? Okay. So I should say, if things do go blurry, please, because I'm not able to see the chat window at the same time as sharing on full screen. So just let me know, okay? Yeah. Could you hear me?
Yeah.
Okay, good. All right. And Vasha, if you can text me, okay, Vasha, if there's a problem.
Sure, Raj.
Yeah, thanks. Okay. So the word development or develop comes from the French développer, meaning to unwrap. And so what I'm going to do is spend some time just talking about the history of embryology. And this will take not so long. And just as a way of kind of understanding how we got to the understanding, how we're at the place we are now. Okay, so even in ancient Egypt, even in ancient Egypt, the Egyptians were aware of the placenta. That's thought to be where the soul entered the body. They were already artificially incubating chicken eggs as well, so they knew that incubation of eggs gave rise to chickens. Probably the next stop on the history of development and embryology was the Garbha Upanishad in 1500 BCE. So and here, so even there, there was quite a kind of accurate understanding of human embryology. So here, you can read this a little bit. On a perfect day and on the uniting of the male and female reproductive fluids, after the lapse of one night, the embryo is in a mixed semi-fluid state. After 7 nights from there on, it takes the shape of a bubble formed out of water, and at the end of the fortnight, it takes the shape of a solid lump. it gets solidified and hardened at the end of 1 month. In 2 months, the head takes form. At the end of 3 months, the legs and foot are forming. And by the 4th month, the wrist, stomach, and the hips, waist, etc. are formed. During the 5th month, the spine and the adjoining bones get shaped. The mouth, nose, eyes, and ears are all formed during the 6th month. So up until now, it's fairly accurate. I think the rest is probably maybe based on cultural references, I guess, yeah. In the 7th month, the life or jivan enters the body shape so far. By the 8th month, it attains full shape and gets fulfilled with all other remaining parts. If the potency of the father is stronger than the mother, it becomes a male, and vice versa. So all of this, so up until the 6th month, I think the people who had done the research for this part of the Garbha Upanishad were pretty good and got it fairly spot on. It does make me think, how did they get the human embryos? And when I start thinking of that, it kind of feels a little bit more icky, I think. Does anyone know the history of this? No? Can you all still hear me?
I had a question.
Yeah.
When we talk about, developmental biology, do we see it as something that like encompasses embryology, or do we see the both the same? Because if we are talking about regeneration, then that will be a part of developmental biology that is not exactly embryology, right?
Yeah, you're absolutely right. So I'll come back to this point, okay? You're absolutely right. For the longest time, embryology was embryology. It was kind of a very descriptive study. But then when we talk towards the end of this lecture, I'll give you the context in which it became the subject that we know now. Okay.
Okay, thank you, sir.
Yeah. So we have our own India contributing to the history of embryology here. After this, probably I'll give a short, brief mention of Hippocrates, who suggested that the conservation of developmental processes between bird and man, and he already had the idea of using model organisms. He has a quote: "Take 20 or more eggs, let them be incubated by 2 or more hens. Then each day from the second to hatching, take 1 egg, break it, and examine it. You'll find exactly as I say, for the nature of the bird can be likened to that of man." So already 2,500 years ago, the idea of model organisms was, the Greeks already had this idea. So Aristotle, who I'm sure you all know, Alexander the Great's teacher, suggested that organisms form from undifferentiated masses. That is, the animal differentiates, its matter continuously proceeds up a hierarchy of forms, where a product of one generation acts as the matter for formation of the next level of organization. So this is a very kind of astute observation of what development is. He already is talking of this hierarchy of forms, this kind of idea of embryonic fields, which I'll come back to later on in some of the subsequent lectures. And so this is kind of very nice, and we'll talk a little bit about Aristotle here. So he suggested that the he kind of put this idea of fertilization here. So he was convinced that the seed of the cock, defined as a surplus product of the blood, forming a foamy mixture of water and vital heat, is responsible for starting the developmental process and for supplying potential form to it. So again, very, as these early guys always were, kind of guys and very male-centric. Once it enters the female body, the vital heat in the male semen passes on its principle of movement to the material inside the egg. Hereafter, the semen evaporates, while the fertilized matter starts to self-organize its potentiality into an actual species-specific form. This is conceived as a gradual process in which the blood is formed first. So remember, Aristotle is looking at eggs, so chicken eggs. He's opening them and looking at them at different times. And the first thing he's able to see is blood. And as anyone who's looked at embryo development, chicken embryo development, such as Varsha, will tell you, the first thing you can see with the naked eye is blood. So Aristotle's idea was that development was epigenesis. That is, it forms from an undifferentiated mass. And that different organs are formed by a cascade of gradual changes in the undifferentiated mass, leading to a well-organized whole. In other words, as the animal differentiates, it proceeds up a hierarchy of forms where a product of one generation acts as the matter for formation of the next level of organization. So this kind of imposes a purpose to development, and it where development is all about the generation of a form, and it also fits in with Aristotle's general teleological view. So Aristotle always believed that everything was for a purpose, and here the purpose for development was to make an adult form. Okay, this leads us very nicely onto the pre-formation epigenesis debate, and I'm not going to talk about it too much, unfortunately. I've written in the notes what I think about this debate and what the history of this is. Basically, as you all probably know, pre-formation suggested that within either the male or female gametes was a miniature version of the adult form, and all development was to kind of allow this miniature version to grow and be nurtured. So this diagram here on the left is very famous. It's the one from Hartzucker, and it's where he thinks he sees a tiny little human in the head of the sperm. It didn't need to be the sperm that was carrying the miniature human. In some cases, the egg also had the miniature homunculus in there. So it took a while for this debate to be resolved. Hans Driesch, kind of almost at the end of the 19th century, probably put it to rest almost completely by this simple experiment here on the right side. Okay, and what Hans Driesch was doing, he was looking at sea urchin embryos, and what he did was to take one of the 4 blastomeres of the sea urchin embryo and separate it into 4 individual cells. And what he found was that each cell was able to give rise to a new embryo, a complete sea urchin form, which seemed to indicate that the gametes did not include a miniature version of the adult, but definitely were epigenetic in kind of Aristotle's sense of the word. Okay. Any questions here? Great. So the I should say that Hans Driesch was in a huge argument about this with Wilhelm Roux, and both were students of Ernst Haeckel. And so you'll hear something about Ernst Haeckel later. Ernst Haeckel came up with this famous diagram on recapitulation theory that maybe many of you have seen once before, even once in this lecture before here. And so Haeckel suggested that every embryo from every organism went through a common stage, this pharyngular stage in the top row. And the more advanced the embryo was, or the more advanced the animal was, all it was doing was adding new stages onto this top row. This, of course, is not quite correct, but we can come back to that in another time. So we know from more modern interpretations of this that there's really probably this idea of recapitulation is probably not quite the whole story. Okay. So at this point, do we have any more questions? Anyone? Can you hear me and see my slides?
Yes.
So,
Yeah.
So,
Okay, thank you. Okay, so epigenetics. So I'm going to come back to now Megna's question about this idea of embryology and becoming developmental biology, and think about epigenetics. And the guy on the left smoking the pipe is Conrad Waddington. Conrad Waddington did more than anyone to kind of start synthesizing the idea of embryology and genetics to make a composite field, which is now developmental biology. He brought kind of a nice nuance to this pre-formation epigenesis debate by introducing genetics to embryology. So a little bit about Conrad Waddington. He's a British scientist. He was born in England in 1905. Soon after, he went and spent the first 4 years of his life at his parents' tea plantation in Wayanad in Kerala, so just down the road. He is an incredibly deep thinker, and some of the ideas that he had were kind of short of genius, I think. Were genius, not short of genius, were genius. He formulated kind of in the 30s, 40s, 50s, and 60s these ideas, and they are still very relevant. And he kind of realized that if you introduce genetics to this pre-formation epigenesis debate, then it kind of starts making a little bit of sense. So he understood that the genetic, the kind of makeup, the instructions that make the embryo are carried in the genes. So the nucleus is kind of like a miniature version of the adult. Yeah, the information at least is in the genes of the parents, and then epigenesis is is kind of the what is it, the asymmetries, the cytoplasmic asymmetries, for example, the mechanical force that the growing embryo experiences to mold its final shape. So Conrad Waddington brought genes into development, and by bringing genes into development, embryology could gain more clarity on biochemical concepts such as the organizer, gradients of signaling, competence, and the embryonic field. And also then genetics would move from this kind of very dry position to being something which has this direct causality, causing, where there's kind of this direct connection between hereditary and variation and to the physiology of descent. So Waddington's contributions were to epigenetics, to gene networks that give rise to phenotype, to robustness as a result of gene networks, and canalization. And I'm only going to go into this briefly. I've put up one review on Conrad Waddington's contributions, which you can all have a look at if you're interested. So this idea of epigenetics, as I said, now combines developmental genetics and this kind of very German term, experimental embryology, Entwicklungsmechanik, okay, which is experimental embryology. And so developmental genetics was being done by T.H. Morgan and Stuyvesant in the US in the turn of the 20th century, and they were studying the genetic function in embryonic development in Drosophila generally. The German school, characterized by Spemann, for example, was looking at these changes in embryo form after chemical or surgical interference. Waddington put these both together, and called it actually causal embryology or epigenetics, and this has now become developmental biology. Of course, all of these feed into things like histological differentiation, regional differentiation, so patterning, and individuation with specification. So all of these concepts Waddington was starting to articulate even then. He came up with the idea of gene networks as well. He understood that the genotype, so the genetic makeup, plus the epigenotype, which is the, of course, epigenetics now means what goes on above the genome to affect the genome. The epigenotype and the environment will give rise to this particular phenotype. And so this is how he was starting to elaborate this idea of gene networks. He took ideas from Stuyvesant on position dependent gene expression and started already elaborating in 1950 the idea of chromatin organization or position of genes within the nucleus as a way of controlling gene expression. And as we'll talk about in subsequent lectures, that's a very important way that we can think about how patterning mechanisms and differentiation regulation happens. Robustness, he conceptualized by thinking about feedback mechanisms. So genes will make proteins. These proteins will affect the expression of other genes, but will form networks where they can feed back on themselves or other regulators to ensure that the expression of genes is robust. And then the idea of canalization, where the potential phenotype of a particular cell becomes more and more restricted over time. And this is something that he famously articulated in this kind of landscape model, which he called the epigenetic landscape. And here again, what we're looking at, I think this is famous. I think many of you have already seen this. Has anyone not seen this before? This epigenetic landscape?
I have not.
I haven't.
Okay.
I haven't.
Even I haven't.
Okay. Okay. So the epigenetic landscape, what you're looking at here is kind of these hills and valleys, okay? And what they represent is the troughs at the end represent the potential path, or potential phenotype of a particular cell. So one trough could be muscle, it could be neurons, it could be bone or whatever. And as this ball, which represents the potential of a cell, what the cell is able to give rise to, goes down the valley, it takes different routes, okay? So the ball, as it goes down, and down is the metaphor for time, as it goes down, it takes this left-hand route, leading to a lineage that will eventually form muscle, for example, okay? So that is an idea of canalization that Waddington proposed. And this epigenetic landscape is underpinned by particular gene networks that ensure the robustness of the cell's phenotype. And we'll talk more about this in later lectures, okay? Because this is kind of a important concept, I think. Just kind of a word here. I'm brought up this analogy of the epigenetic landscape. You know, these hills and valleys, and it seems like always we're using. I mean, this is just an aside. We use analogies to kind of make sense of what we're learning, and research is anyway about somehow breaking those analogies sometimes. So thinking about different analogies as well. And so at the moment, I'm directing you to think about the potential of a cell in terms of these valleys that the cell is traveling down into, okay? There are probably different analogies that you can think of once we've done these series of lectures that more accurately represent what a cell can give rise to based on our current understanding with things like induced pluripotency and those kind of terms, okay? And so I think one thing we should always try and do is not be too complacent with our analogies. Okay, so these are Waddington's contributions, okay? There are reviews out there, and there's one or two in the Moodle reading list, which go into much more detail about Waddington as well, okay? And so you should have a look at that. I'm going to finish off this lecture just talking about what we're going to try and learn in the subsequent lectures, okay? And so what you're looking at here is the development of Xenopus, I hope, yeah, here, okay? And you can see that the first thing you see is cleavage, so cells are dividing. They started dividing kind of in a synchronized fashion, but then lost their synchronicity. And now what you'll see is some changes to the pigment. It looks as though the cells are moving, undergoing morphogenesis. And then, fortunately, and then you'll start seeing the neural tube starting to fold and come together. So this is Xenopus, and you can see this kind of gradual emergence of form from the single cell. Okay, here it's now trying to become a tadpole. You can see the cement gland here. The eye is here, and it's now growing, and it will hatch from its vitelline membrane, and here this ugly creature at the end is the final product. Drosophila as well, you can see this in very clearly. Again, this is looking at the nuclei of the Drosophila larvae, and you can start seeing that again, division is synchronized, and then you undergo this dramatic morphogenesis, desynchronization of division, and the gradual elaboration of the form. Okay. This one we've seen already, the zebrafish embryo with what's going on in the cells as well. Okay. Yeah. So I bring up these 4 videos, and remember the videos. So we need this element of time to understand where a cell has come from and where it's going, what a cell started doing and what it ends up doing, what a cell was and what it has become, to really understand fully what developmental biology is. And so the concepts that we do need to understand, and I'll try and explain to you, is cell division, how cell division is controlled, and we'll talk about that in the next lecture. The emergence of pattern, that is pattern formation, which involves the generation of axes, germ layers, processes such as induction and lateral inhibition, which all come from signaling interactions. And so these we'll spend some time on. The words that may not be so familiar to you, such as germ layers, induction, lateral inhibition, we'll explain. And anyone who still doesn't understand them, we can talk about them in more detail. Then we'll talk about morphogenesis, which is how we get a change in form. So how do cells and tissues rearrange to form new structures and new shape? We'll talk a little bit about cell differentiation. Cells will form many, many different types of cells. And so how do they differentiate and what does that entail? And here we can go back to cell cycle control, and we can also think about what happens to the genome when cells differentiate. We'll talk a little bit about growth as well, and how tissues, organs, and organisms increase in size. We'll also try and touch on evolutionary developmental biology, and so thinking about how variation can be introduced into these different types of form as well. And then talk about the techniques you can use to understand all of these processes in the modern time. So those are going to be my lectures. After me, Sandeep will talk about kind of the basic physics behind some of these patterning and signaling events and morphogenetic events, before Tina will talk more into the genetics of developmental biology. So that's kind of all I wanted to say. I'm going to stop sharing. I'm going to start my video, and I'm going to ask if there are any questions. Can you hear me?
Yes.
Yeah. So if anyone has any questions, please feel free to ask them. And don't be afraid to ask any question.
Raj, could you elaborate on robustness of gene networks, what that means?
Yeah, so what that means is that the gene network is somehow insulated from any perturbations. So there are 2 concepts I want to kind of highlight here. The first is this idea of feedback control that ensures that the level of a particular protein is always the same, no matter what kind of stochastic events are happening around the gene. So for example, the temperature of development is not quite optimal, nutrition is not quite optimal, but the gene is always expressed at the same level at the same time, is one idea. And then another idea is that there are kind of these insurance mechanisms. And so Spemann called this double insurance, that one gene network will, for example, give rise to the eye. But there is also a redundant gene network that will also give rise to an eye in case there is a mutation in the first. Okay, So essentially, these are ways to buffer the embryo from mutation, from environmental challenge, and from any kind of toxicity that the environment or either the kind of general environment or the maternal environment may be subjecting the embryo to. Is that kind of clear?
Yes, got it.
Yeah?
Yeah.
Okay. So any other questions?
Hi, Raj.
Hi.
Hi, I had a real question. You initially mentioned that development could be defined as a consistent increase in complexity from like single cell to multicellular organism.
Yeah.
Are there some there's some organisms where like they develop and become like, so for example, the anglerfish, the male is really small and not complex compared to the female.
Yeah.
So is there some point in the developmental like transition where it becomes more complex and then becomes less complex? Like,
Yeah. So there is, yeah. So Nishan, that's a good point. One of the classic examples are some species of ascidians, so some species of tunicate, kind of basically start off their larval form quite complex, and then when they become an adult, they digest their nervous system and just settle onto a rock and feed. So there is kind of this idea that generally in metamorphosis, some species will lose their complexity.
okay. So it's not the definition is still up for debate.
Yeah. The definition is still very woolly. Okay, Girish.
Isn't complexity subjective?
Yeah.
Then
Absolutely.
how do we use that to define certain things like what we just did right now?
Yeah, tell me.
I have no clue.
Tell me. I guess we have a sense of complexity. Yeah. That there's more than several cell types, for example. That there's a kind of particular organ systems that are somehow working together. But you're right, complexity is subjective, and that I think was one of the problems for the definition as well. That complexity is, what is complexity, and there are species, Volvox, for example, a kind of a colonial alga, lives as a ball of cells, essentially. It undergoes various morphogenetic movements. It can be thought to be developing, but it has very little complexity. Colobacter as well will divide to give a free living swimming form and a reproductive form. There's very little complexity there, but it is developing as well. So it's all, the definitions are woolly, and maybe it's okay for them to be woolly. There's if you have a look at that Stanford philosophy dictionary definition of developmental biology, they make an argument for they kind of explore the idea that there's no theory of developmental biology, and their conclusion is it's okay, and not for the reasons we think that it's okay. And I'll let you all have a read of that. Okay.
Yeah. Oh, and
Hi,
yeah.
Okay.
Go ahead.
Okay.
hi Raj,
Hi, Anju.
Anjum here.
Hi.
I wanted to ask, you just said while explaining robustness that nutrition of the medium won't affect genetic expression of proteins. But if that does not affect, then when the nutrition is not available, won't the cell undergo stress while continuously producing the proteins that are required for development?
Yeah. So Anjum, you're absolutely right. So when I talked about things like nutrition, these are of course I'm thinking more of minor fluctuations rather than more acute situations where there's absolutely no nutrition. So I think robustness ensures some level of buffering, but of course, when things get too acute, then no amount of buffering will help the embryo there. And so it may be a question of kind of understanding what that tipping point is.
Thank you.
Okay. Okay, Garish, you had another question, or you're okay?
Yeah. Are we not going to touch plant developmental biology?
So I need to ask Shiva to do some plant developmental biology sessions. Plant development is fantastic. I'm unfortunately an animal developmental biologist, but plants also develop, and they develop fascinatingly. And so in the past, we've had Shiva give 3 or 4 lectures on plant developmental biology. As this semester's a little bit truncated, I haven't asked him, but if there's kind of a feeling that we'd like to do plant developmental biology, I'll ask him. Okay.
Yeah. Sure.
Okay.
Thank you.
Okay. Okay.
Raj, you also mentioned about development in unicellular organisms.
Yeah.
So could you say more about that?
So I can, but I am not very good at it. Okay. So there are bacteria, for example, that will differentiate into different types. Yeah. So I mentioned Colobacter, which under particular stress conditions will give rise to a kind of a swimming, free living form and a reproductive form. So this is kind of development, if you want to use that definition. Volvox as well, which lives as a ball of cells, so it's potentially unicellular, but it can aggregate as a colony, and it undergoes morphogenesis as well. Next week or the week after, I can't remember which one. Oh, sorry, next lecture or the lecture afterwards, I'll also talk about Dictyostelium as well, which is a classic unicellular organism which can become multicellular as well and develop very beautiful phenotypes as well. Okay.
Yes.
Is that okay?
Thank
Is
you.
that okay?
Yes.
Yep.
Yeah.
Cool.
Hi, Raj. Again.
Um,
Sorry, I was just thinking about the analogy of development where it's like a ball rolling down a hill, which is similar to the way that people think proteins fold and whatnot. But
Yeah.
I thought that it may not be like, I thought maybe a better analogy would be like a rocket propelling or something, because all the energy already exists. There's no energetic minima in development, right? Or like, are they going towards sort of energetic minima or something?
Not really.
So then why is that analogy useful? Because
Yeah, so I think, I mean, the idea of the minima is kind of potential minima. Yeah, so to minimize the amount of work that this cell is having to do. Yeah, and kind of being potentially open to many different fates. Yeah, and by the time it gets to its destination, it can basically turn off a lot of regulatory mechanisms and just concentrate on being a muscle cell, for example. I do need to kind of understand this a little bit more. I know that NCBS will have a new faculty, Achisman Raju, who is looking at these kind of actually this epigenetic landscape and trying to understand more about these kind of concepts and what the minima means in the epigenetic landscape.
yeah, thank you.
Yeah, it's a good question.
Yes, even I had a follow-up question on that analogy.
Uh-huh.
So, like, the cell is basically the ball which is rolling down that hill, and
Yeah.
there will be some topographical like differences between various hills. So,
Yeah.
and then we talked about separate hills are being regarded as some of these to be the muscles and then some to be bone, then when the cell is rolling down, so there could be some, is there something some difference with respect to how the cell develops into a muscle or into a bone, some like energetically differences which have been talked about, like the topology?
So, the actual route that the cell takes will be dictated by the kind of the environmental signals that that cell will experience as it rolls. Okay, and so the cell will be as it rolls down the valley, it'll be subject to particular signaling molecules, for example, particular types of nutrition, whatever epigenetic actions that the cell experiences will dictate which of the troughs it will roll into. I know it doesn't quite answer your question about minima and energy, because I think that this may not be a strict analogy. Okay, so it's more of a metaphor. Okay, of what the potential of the cell is. Okay.
Yes, sir.
Okay.
I had another question. Do we consider all kinds of cell differentiation to be involved into developmental biology? Is that what it's about? Cell differentiating into different specific cell types?
Yeah. So definitely, I think at a minimum, all cell differentiation is part of developmental biology, and all cell differentiation, I think, uses genetic networks to enable that differentiation process. Atal?
Thank you.
Yeah.
Like in plant biology, the plant development biology, we see that there is the special ability with plants that they can de-differentiate their cells back into the callus form, like
Yeah.
we do in plant culture.
Yeah.
So doesn't that go against the developmental biology definition? Like the plant cells are getting de-differentiated and then moving back to getting differentiated.
Yeah, I mean, animal cells do, or animals do the same thing as well. So when you pull the tail off a lizard, for example, it will form a blastema, which is kind of a de-differentiation event before forming reforming the tail. I think this might be what I think Nivedita was saying about kind of regeneration and repair, probably needing to be included in this definition as well. Yeah, because I think the callus also is a kind of repair process.
Yes, sir.
Yeah. Okay. More questions? Anyone? Okay. Well, if there's no questions, I'll let you prepare for Neurobiology 1A, which is coming up at 11:30, I think. If you think of anything, then let me know. I'm going to put up a discussion board in the Moodle page as well. So whenever something interesting crops up or something that you feel you want to discuss with the class crops up, then just add it onto there, okay? So with that, I'll see you all on Wednesday at 9:30, okay? Yeah. Okay.
Yeah.
Okay.
Thank you, sir.
All right. See you. Bye-bye.
Thank you, sir.
Thank you.
Bye.
Bye.
Thanks.
See you.
Thanks.
Bye.
Bye.